Okay. Thanks for the kind introduction and hello. I'm Anand. It's a great pleasure to be here. So thank you, Kyan, for the invite to also bring me to your lovely country, meet old friends and some new ones, hopefully. Um, Clarification, I'm a medical oncologist, so I treat lymphomas, but I don't know anything about leukemia treatment. But I can sort of clarify some things about how cell-based assays work. So if we want to discuss the interpretation of these assays at the cellular level, I can certainly talk to that. I also have a lab at the Cancer Science Institute, which is opposite the hospital. So and that's how I got involved in the study, because the lab next door to mine was Ed's lab, and that's where we got talking about this. So that's the background to this work. So disclosures, the most important and relevant disclosure for today is that I'm I'm actually an inventor on the patent that is involved in, which was licensed to Kyan, largely because I helped Ed do it as a clinical collaborator. I'm here as a guest of Kyan to speak, but I'm not involved in the running of the company or in any of the company's operations. So I'm at liberty to tell you all the strengths and limitations of these approaches. So feel free to ask about these things. I'm a practicing clinician, just like all of you. So my role has been only at the level of the initial stages of developing this and bringing it to the clinic, but I'm not involved in its commercialization in any way. So we're a small group, so I don't think we should be formal. Please interrupt me at any time. I'll try and be as sort of, the idea is to actually tell you a bit of a story behind how this came about. It is a Friday evening, and so, we don't want to be too heavily scientific, so I'll kind of tell you a bit of history of this QPOP test as opposed to actually the deep science behind it. So I think we all know the sort of the concept of this personalized medicine as opposed to generalized medicine that we practice. So we also were interested in that. But I want to take you back a couple of years to why this is not so common. And the reason actually is there were guidelines that came out in the 2000s and later in 2011 on actively saying don't do functional precision medicine in oncology. This is what the ASCO told us. And the reason for this is that around in the early 2000s, there were lots of studies that actually tried to take out patient samples, test different drugs on them. And many of them were very poorly run, but also poorly predictive. Now, at that time, most of the drugs that they were testing were chemotherapy. And most of the ways that they were testing it were somewhat crude. They were MTT assays. And so things have actually changed quite a lot since then. So since then, we actually have quite a bit of evidence, mostly in the space of hematological cancers, that you can take uh, cells out of a patient and you can put it in a plate and you can see that potentially there's some relevance to what may happen in a human being. But changing something like this will take many years. So let's say this is the background at which we are coming at this. Um, so what do we define by the space called functional precision medicine? Is essentially we know precision medicine as a way to define patients based on narrower and narrower molecular markers or some sort of test. But the functional word comes by saying, can we take the cells out and actually check a readout of how the cells are functioning? And that's difficult to do. So the idea is, how do we try and do that at a better level? So you can see that there are everything from, could we do it in a mouse? Could we do it with microfluidics? Could we do it with just cells kept alive? The simplest one is just this: just keep the cells alive, low cost, and that's what actually QPOP or now Optim.AI is based on. So there were some trials that were done by some of our colleagues in Europe, which gave us a bit of impetus to go on this. But I must say, in truth, we didn't wait for this trial to start. We actually started even before that, and I'll tell you how we started. Um, the background is that we all know, as practicing oncologists and hematologists, that one drug is often not enough for cancer. You need many drugs. So, and a lot of these early studies that I was telling you, which informed the negative ASCO guidelines, were all one drug based, and it was trying to see whether you can put higher dose of one drug to actually kill a cancer cell. But really, ideally, we want a situation where we can get many drugs together. So Ed, who was at that time at the CSI, he had just published a paper showing this, taking this sort of principle of many biological systems follow this sort of a simple quadratic equation that actually leads, that allows us to predict the overall response from just a few data points. Meaning you don't need every point on this curve, but you need a few points and you can predict the rest. So what that allowed him to do was to take that to study combinations, with a small number of cells, and he showed this in a mouse model of multiple myeloma. And he was very happy that his lab published a Science Translational Medicine paper, and so I went out with him to celebrate this, and we were having a beer, and we were talking about the fact that I had a patient in the ICU at that time with a refractory gamma delta T-cell lymphoma, and you all know this is a terrible disease. This was a patient primarily managed by one of my colleagues who was his primary physician, but I was involved in the care. And the patient had a white cell count of about 300,000 at the time, all malignant cells. And we had plenty of cells to go by. And we already had an ethics protocol that allowed us to collect these samples. So I told Ed that day, I have a lot of cells. Why don't you try it in lymphoma instead? You've done this in myeloma. Myeloma is so rare. The patients are so old. So let's do it in lymphoma. And so we decided to take this one patient, and we actually tested it. And it came out with this combination. I helped him pick a set of drugs that we should try. Some of them standard, some of them somewhat new. And we chose bortezomib and panobinostat because there was a phase 2 trial that was run from Singapore on that. So this particular patient had a very unique sensitivity to this. We treated the patient, and his counts just plummeted very quickly. Pretty much, almost walked out of ICU by the time that he was ready, and he responded remarkably to this. Unfortunately, after his autologous transplant, he went back to Brunei and then couldn't come back to Singapore during COVID, and he actually died later. But it was a very good response overall for this bad disease. So this kind of, sometimes you have beginner's luck, and this was our beginner's luck with this study, because after this, the next few patients that we tried didn't work. What do I mean by didn't work? We got samples, we sent it to Ed's lab. This was before the QPOP protocol that we used to call it was even set up. It was just we were just using one of our other trial protocols at the time. Study protocols. But we didn't. In the beginning, we were just trying to see how do we choose between different salvage chemotherapy regimens. So we were going GDP, ICE, DHAP. How do we choose between these things? And it was not very good at coming up with significant differences from patient to patient. So eventually we set up a sort of national study for this whole process, where we could go from the patient biopsy, or if it was liquid, then you have to do a sorting step to take out the tumor cells, do the test, come up with combinations. And my role really in this was, which drug should we put into the test? How do we interpret the assay? How do we depict the assay so that clinicians can understand it, and then go into an actual testing? So this was my input into the early development of QPOP in non-Hodgkin lymphoma. And really, this was the first human study that we did in this disease, using this technology. So with this, we actually ran a national study, quite a lot of patients at that time. We were able to refine how long it takes. In the early stages, it used to take close to 2 weeks. Ed's lab were able to bring it down to about 1 week for the initial testing with that. So that was a big, important difference. And you probably have seen some of these reports. This was something that me and Ed developed, because the first version of the reports looked like this. And uh, he just gave a map like this, and all I put it up at our tumor board, and all our clinicians were like, "What is this? There's a pyramid over here. I don't understand anything." So we had to convert it into something that clinicians could sort of feel a little more comfortable with. So what we decided to do was, clinicians like forest plots, so let's make a forest plot. So that was our first aim. So to say, okay, more to the left, it is good killing. There's a line in the middle, so we can at least see how far we are from the line. So this was one part of the thing that we did. The other was that we wanted to pick doses of the drug that are very far away from what your Cmax is. So meaning it has to be something that we can actually achieve in a human being. So the in vitro test has to be, so 15% or 30% of that almost. So much lower, so that you can actually achieve these doses in the person. The other was, which drugs do we actually put in? And then how do we present this in a way that you can, because in each thing you have a high and a low dose, how do we then see how these, the more blue it is, the more dead it is, and the more red it is, the more live it is. That was a sort of simple way that we thought of showing this information. And this is, I was telling Kyan that they need to evolve past this, because this is now 10 years old. You need to come up with better and cleverer ways of showing this. But this was what we originally developed to be somewhat clinician friendly compared to the old version. So with this experience, this plot really says that we saw what we expected to see, which is that actually the things that worked most were not chemotherapy combinations, but typically a combination related to a targeted agent and a chemotherapeutic. So in our experience, what we discovered was that the initial cases failed because, or rather were not useful because we were only looking at chemotherapy. And this is an important lesson for how we use these things in the future. We need a variety of drugs with different mechanisms of action, which all work at the cell intrinsic level, meaning that you don't need the immune system for it to work, for these types of first pass assays. Now I know Kyan has got the new version which includes T cells and peripheral blood cells, but we can talk about how that will work later. So B cell lymphoma, a PI3 related drug was the strongest sort of partner. In T cell lymphomas, romidepsin was the strongest partner. So expected. This is expected information. So we were, so that's useful to know. We had some patients that we could treat based on this approach. Not all patients, because for us also, just like you, many patients actually cannot afford the off-label treatment. If it is on-label, it is easier to do, but if it's off-label, not so easy. We treated some patients and we saw some complete responses in unusual situations, as I hear you all have as well. But we also learned about how not to do this, because when some of my colleagues who were not so familiar with giving a new combination, if you give it and you just monitor the patient 2 weeks later like you do with a standard combination, sometimes in between you have a significant sepsis event. So it needs almost a monitoring like you do in a phase 1 trial. So that's another important take-home point for how we are using novel treatments. You have to monitor them like you do with any novel trial. And so that's something to just bear in mind. How do we know that these things are working? We with these types of studies, if you have enough data points, you can compare it to a whole group of patients receiving standard of care. But we don't have that many patients there. So actually the best comparison is to the patient themselves. So the patient's previous line of treatment, how long did it last, to the patient's current line of treatment, which is QPOP based or precision oncology based, how long did it last? And that's called a PFS ratio. So that's one of the ways in which one can formally study this, which is increasingly being used in the literature. So that we show that if it goes above this value of 1.5, that means that you're generating clinical benefit. And we do that for many of our patients. So here are some examples. So this is one of my patients who had a refractory DLBCL. The prediction actually showed quite a lot of resistance to most chemotherapeutic combinations. sensitive to everolimus and palbociclib. We were not brave enough to give her that directly, so we gave GDP, and as expected, she had no response. And then we gave her this, and she actually had a complete response. And she, I treated her now almost, I'm tempted to say 6 years ago, and she's a DLBCL who only received about 6 months of this double treatment and then just some palbociclib, and she's still treatment free to this day. So it's a unusual, long-term remission that she achieved. No bispecific, no CAR T, nothing like that. I saw her just last week. And this was T-cell lymphoma that one of my colleagues from the private sector, he treated. It was a NKT. This patient had a very good response, but unfortunately progressed afterwards and then passed away. So this was the sort of more typical response to an aggressive lymphoma, but at least a complete response for a significant period of time. This is my patient Peggy, who is still alive. So this was our initial work, which we published, I think, about 3 or 4 years ago. That was about 70 patients, our initial sort of study. And this year we published the full follow-up of all our patients, 120 patient cohort. This is Ed, by the way, for those of you who probably have never met him, and this is me with hair when I was still doing these studies. I'll tell you a little bit about what we have learned from the full combination study, that the overall results of this. And please feel free to stop and talk about specific points in between. And so I was telling you how we evolved the drug set as we went along. So you can see lots of drugs over here, different classes, chemotherapy, non-chemotherapy. Initially, we were mixing and matching a lot of things. We were trying to, every patient was a slightly different experiment. And eventually, we settled on a more stable drug panel. What did we learn from that? We learned that we should have combinations of drugs which have different properties, because it is that shows the difference from patient to patient. So that's one major message to take home. If you're building a panel, try to make sure that you have drugs with different classes in it. In our overall study, this is the evaluable patient group. So we had about 120 patients, of which maybe about 10 of them, the QC didn't work. Another 10, we didn't actually have enough the patients were lost to follow up. We couldn't do this. So this is the total number that we have for the study, about 100 patients. As you can see, most of them are DLBCL from B-cell lymphoma, and many of them were the typical subgroups, PTCL, NKTL, AITL, for the T-cell lymphoma subgroups. So we do have overall this consistent more than 50% complete response and partial response for those patients that are treated with a new treatment defined by this platform. But bear in mind, this is only 27 out of a full group of more than 100 patients. What happened to all the rest? They received standard of care treatment because they couldn't afford it, or because they were not willing for it, or whatever reason. But within that group, one of the ways that we can compare it is this. I was telling you this comparison of the PFS of the same patient from the last treatment to the next treatment. And so in that, what we see is that, generally the patients that received a sort of guided treatment approach had a higher PFS ratio benefit. And we see that in this sort of Kaplan-Meier as well. This is a sort of, I would say, a less critical plot, but it's from a diagnostic labs point of view, it's important, which is what is your actual the ROC of this test. But the way that I would think about it, actually, if you're interested, please look at the study. I won't bore you with all the details, but I'll tell you, as a clinician, when I look at it, we only want two values. What is the positive predictive value? What is the negative predictive value? Because you're treating one patient at a time. Kyan is making the test. So for them, sensitivity and specificity are important. For us, PPV and NPV are important. The highest value is really for the NPV. And I highlight this because this is a general feature of many ex vivo tests, is that they are actually the strongest value may be telling us what not to do. what are the drugs that we maybe should not be giving a patient. This is actually an underutilized value of these tests. Of course, as clinicians, we're looking for new options that we can give our patients, but sometimes it's also equally good to see how do I use this to refine current options to make sure that maybe I shouldn't expose the patient to all this toxicity. In fact, it is usually or in all ex vivo studies, the NPV is typically higher than the PPV. So 70% PPV is not bad, but the NPV being 80%, I think, is even better. So that's one important takeaway from a clinical perspective. Since then, my colleague Daryl, who in the private sector has assembled a small set of T-cell lymphomas that he had treated, and he's kind of he presented this at IHA last year, and in that also again we see a sort of similar rate of a significant number of patients with CRs and PRs using this kind of approach, even in a situation with refractory T-cell lymphoma. If you look at the types of treatments that he gave, many of them you recognize. They are often things that actually may even be standard, like romidepsin, in the sense that it's approved in that setting. But there are situations where they've seen responses to venetoclax-based combinations, bortezomib-based combinations, AZA-based combinations, so other ways in which you could use these drugs in relapsed refractory T-cell lymphoma. So, and this has been our experience as well. So ultimately, even in our full patient study, it is still copanlisib-based combinations that rule. This is again, I highlight that this is the first version of the test where we don't have the immune system. This is just the tumor cells, and we're trying to see what kills the tumor cells. So with that, these drugs still sort of are the preferred combination partner with other chemotherapeutic drugs, but it doesn't mean that you can't use something else. So a few sort of nuances to note from this. We had a few patients who had multiple tests, meaning early in the disease they had one test, and then when it relapsed they had another test. So those patients are useful to tell us how stable the platform is. And actually the answer is it is quite stable, because the most things fall along the diagonal. There's just some combinations that are seen in one versus the other. And indeed, some of them were related to, for instance, the patient was treated with venetoclax, so therefore they were resistant in the later line. Similarly, in these, most of them are actually still along the diagonal, and there are again these venetoclax samples combinations that come up as more resistant, and that's actually because even the resistance mechanisms to DHAP and CAR-T, some of them relate to these BCL2 pathways, these apoptotic pathways. So it is unsurprising that actually they do have cross-resistance to venetoclax in this setting. This is a complicated plot, but actually a very important plot, and I'll explain to you why. It is about some drugs are essentially very dependent on the dose in the plate. So what do I mean by that is there are some drugs that the killing will actually go up as the dose goes up, always. And the frequency where they occur in combinations will go up as the dose goes up. What does that mean for us? You have to take these combinations with a pinch of salt. So if you see a venetoclax-based combination, you see a vinorelbine-based combination, you see a bortezomib-based combination, you have to think twice about, and I'll show you, they may always rise to the top because these drugs invariably will work well in a plate because they're highly, highly potent drugs. But they may not work well in the person. But there's still a way to dissociate that, and I'll tell you how we can potentially dissociate that. There are some things that are unrelated to this. So I think at the end of that, some of the considerations that we kind of want to know are how much of the combination is governed by one drug and not the other drug. How heterogeneous is this within a patient or within a group? Ideally, we'd like the same patient's blood as a control, so that you can kind of compare and see how does it kill abnormal cells and normal cells. This would be a thing for the future. And how do we scale it? And scaling is what Kyan is doing. So this is, generally the principles that we're interested in. These slides I got from Ed. So after our study, this was licensed out to Kyan. They've gone ahead and done it. I was explaining to them, I think of this like a child that has left home. Now I only want to see you at Chinese New Year. No more. I'm not doing much work with them anymore. But I remain interested in this because I was involved in it at the early stages. And but what we have done is I've helped our team develop a clinical trial protocol that has gone past this. So it's actually led by my colleague Sanjay, and I'll explain how our clinical trial works. Some of the considerations for using it are who will pay for the drugs, because I'm sure you struggle with that as well. You may find a nice drug, but who will pay for that to be used? Because a clinical trial to do many different drugs, as you can imagine, is extremely complex. So we ultimately designed a trial like this, and it's finally started. And potentially in the future, maybe we can open a site in Thailand as well, if you're all interested. But this is something that we are doing right now. T-cell lymphoma is our main focus area because, as you know, B-cell lymphoma, we have CAR-T, we have bispecifics, we have many things that work well. T-cell lymphoma, we still don't have those things. And we see still good responses in T-cell lymphoma patients with unusual regimens, in this case, a venetoclax-based regimen. So we initially tried to look at, these are just cell lines of T-cell lymphoma. We came up with many drugs to see which ones are highly killing them, which ones are not killing them at all in a plate, which ones are variable. And the variable ones are the interesting ones because that's what's different from patient to patient in a sense. So then we build a panel based on that, and that's what we ultimately put into our latest version. And I was telling you, I'll tell you how to interpret whether a drug that works very well in a plate should be used. So let's take a drug like bortezomib. In general, it will kill cells because it is a very potent drug. But you can see each dot here is one patient or one, and you can see there's a wide variation within that group of patients. So that is actually important to know. And it is important to know, even if for this particular patient, it may be that bortezomib is the best drug of all the other drugs, but it doesn't mean that it's a good drug, because actually what you need is this level over here. So and this is a nuance that you might need to in discussion with Kyan, try and understand where does this patient stand among all the patients that we have seen. And that will help you to choose sometimes a more rational way of using some of these more expensive medications. So this is the actual clinical trial. We call it CombinXT, which is combinations ex vivo in T-cell lymphoma. It's run by our Singapore National sort of Cancer Consortium, so it's got a STCC number. My colleague Sanjay, I've handed over QPOP and Optim.AI to him, so he's now doing most of these things. And this is the protocol, which is essentially, we have a large panel. We choose 12 of these drugs, so that's a unique design in itself. Then we do the analysis. If you're doing an established combination, you just use your standard dose level. If you're using a new combination, we always start with 75% of each drug because we don't know what it's going to be like. We give induction therapy, and then we decide if you receive a CR, do you go to transplant? If you have a PR, what will you do with maintenance? If you have SD, what will you do? So this is the general principle that I would recommend anybody who's using novel doublets do this, which is to use 75% of each drug. This is the sort of turnaround time it comes to us. We actually have a core team within the trial who look at each patient's report, and then we say, this is the sort of group, so from all the key hospitals and myself, and we say, okay, we recommend this particular drug combination for this group of patients. In the long run, ideally, you'd want to take humans out of this and maybe AI the whole thing, but for now, it is still running on human work. And then we start treatment, hopefully, within 2 weeks. This is very important which is I was mentioning follow-up for these patients and how we monitor these patients. Because if you're using a new doublet, we should try to think how do we do it on a phase 1 clinical trial. So typically we see the patients weekly. We may do labs twice weekly. Think of it like you're managing an epoch, or something like that, where you want to manage the patient with some close monitoring. So that is the kind of approach that we would do. Where do we go next? I mentioned that this whole platform was initially designed to be just about killing tumor cells. But we know that the immune system has a big role to play. There are many drugs that work with the immune system. So how do we build that in? Bispecific antibodies, monoclonal antibodies that work with ADCC, ADCP. So I actually had a PhD student that I co-supervised with Ed, and she came up with a new platform to help incorporate. So she generated a modified NK cell line to do ADCC, and so she initially did the first version where she set up a high throughput microscopy assay, which then Kyan have taken on to build Optim.AI 2.0, where you can do both a modified NK line to do ADCC, or you can do the patient's own peripheral blood to do either ADCC or even T-cell engagement with a bispecific antibody. Does it work? We have preliminary data that it does. This is a monoclonal antibody with just, you can imagine the effector NK cell. And what you're seeing here is more cell kill as you go up like this. So only when you add the NK cell and the monoclonal antibody, we see the cell killing. And this cell line doesn't have the target, so there's no cell killing. And similarly, this is a reverse of this graph. So if you see it going down, that's the viability going down, and it only goes down when you have the bispecific antibody and PBMC to cause the death. So it does work in that sort of setting. And we spent some time actually optimizing this. I gave Ed quite a hard time about this because we wanted to see this difference over here, because many studies actually don't show the difference with PBMC and without PBMC, because bispecific antibodies will kill cells by themselves just by direct activity. But what you want to see is that huge difference when you see the PBMC present over there. So that was, and this is now incorporated into their assay. So hopefully it will help in the future with these. Also, because bispecifics are so expensive, it will be helpful to know who not to give a bispecific to. So I think that's going to be an important area. So with that, I think this is where Kyan's part is, where they've gone from the first version, which we helped with many years ago, to the newer version, which includes all these things. I haven't done anything with checkpoint inhibitors. I've only done ADCC and bispecifics with them, so I can't comment on that. But at least in those two settings, I have some sort of confidence that the basic platform seems to work. So I think I'll stop talking here, and hopefully we can have a bit of discussion. That fundamentally, putting cells on a plate and testing what happens to them is a concept that can be considered, I think, now. In fact, the pioneer of this concept, Anthony Letai, is now the head of NCI. He's Donald Trump's appointee as the head of NCI, but he's actually a very good scientist. Many years he's been doing this. He's been quite meticulous about the whole thing. And I do believe that there is merit to this approach. It's important to understand this IC50 dependence. What I mean by that is the in vitro efficacy versus the in vivo efficacy, so that you use that accurately. And I can, as you share your cases, we can reflect on this a little bit. And then you have to have phase 1 like monitoring. I think that's important for patient safety. So, and then ultimately, how do we do all these new drugs in this setting? I mentioned that, this was my earliest work. Now I do more spatial biology work. I mean, I've always done spatial biology work, but this is my current work. And this was Ed and I in our younger days when we started off all this work. And these are our collaborators, our funding organizations. And it actually started, this whole work started with this little seed fund. It was not a big grant. We actually had only $30,000 to start this work, and now it has grown into quite a lot. So it's been a interesting journey over the last 10 years, and now I'm happy that Kyan have taken it on and they've grown to where they are.